Last week we were talking about our special senses. We got through smell and taste, which we learned were two very similar sensations. Unfortunately, our next sensation is very different from those. Okay. Our next sensation is audition or hearing, which takes place within our ear. Now, when you hear about your ear, you think about your ear. Generally, what you're going to think about is what you can see, what we would call our outer or external ear, which functions to funnel sound waves into the middle ear. Okay. Your outer or external ear includes what you can see, the ear itself, and then the canal that carries those sound waves, what we call the external auditory canal or acoustic meatus, to the eardrum, which is known as your tympanic membrane. Okay, tympanic membrane is going to cover the opening to the middle ear. And your middle ear also includes three bones, or auditory ossicles, malleus, incus, and stapes, or hammer, anvil, and stirrup. What they do is basically transmit sound waves from the eardrum to our inner ear. What is pretty cool is what happens. Those sound waves that are currently resonating in your inner ear are first traveling through your external ear and striking your eardrum. And then it basically sets off a domino reaction where the malleus rattles, the incus rattles, and then the stapes rattles. And then fluid inside of our cochlea rattles. Okay. The cochlea is our inner ear, which is where transduction of sound waves occurs from sound waves into action potentials. And therefore, then action potentials are traveling from our cochlea by way of the cochlear branch of our vestibular cochlear nerve to our brain. And we'll get to that in a second. We'll see also here in a little bit that the inner ear, kind of more over here, is where we have cells that create action potentials that are relating to the sense of balance or equilibrium. Okay. Now, in order for all this to occur, we first have to transfer those signals inward. And that's where your middle ear is really going to do the buffering and amplifying of the sound waves. Okay. Our tympanic membrane, or eardrum, like I said, covers the opening to the middle ear, has 18 times the area of the oval window, which is that opening to the inner ear. This difference allows the energy of a vibrating eardrum to be concentrated to a very small area. So we're really pinpointing it inward to a small area. Your ossicles or ear bones serve at times to decrease the transfer of energy to the inner ear. This decreasing energy act is called the tympanic reflex, and this is due to the small muscles that attach to the ossicles and can contract to reduce the mobility of the stapes. Now, unfortunately, this reflex has a latency or delayed period of about 40 milliseconds. It's not quick enough to protect our ear from a sudden artificial noise like a gunshot. And that's why your ears will probably ring after you hear something loud like a gunshot. But other than that, we kind of absorb the shock coming inward. Now, within our inner ear, inside of our cochlea, are hair cells, which are the receptor cells for hearing. They have the capability to transduce mechanical vibrations into electrical signals. And the region of the cochlea that you find them in is a organ of Corti, or spiral organ, which is found in one of the three chambers if you were actually to open up the cochlea. Okay, if you've taken anatomy, you got to see these. So we don't really remember it in great detail because we didn't talk a lot about it. But there were three chambers within each cochlea, and the cochlear duct is the chamber where you find the organ of Corti, or spiral structure. At the tip of each hair cell are 40 to 80 stereocilia, which are little hair-like microvilli to the actual receptors for sound. These hair cells will synapse with first-order sensory neurons that form the cochlear branch of the vestibular cochlear cranial nerve. Okay. The picture on the back here, the previous page, shows this a little bit better. The vestibular cochlear nerve is cranial nerve, I believe it's 8. Okay. Half of it goes to the cochlea, half of it goes to the vestibule. There's where the name vestibular cochlear nerve comes from. There's a vestibular branch and a cochlear branch. For hearing, we're concerned with the cochlear branch. Okay. The first order neurons synapse in the medulla and terminate in the midbrain. Some of the cochlear nuclei decussate or cross over and terminate in the midbrain. Others don't, and they terminate in the pons on each side of the brain. So here we're showing this occurring. Coming into our medulla. Some of them are crossing over, as we see here. Some of them are not. Okay. Both signals coming from the pons in the midbrain are going to eventually get conveyed to the thalamus. What's the nickname of our thalamus again? Gateway to the cerebral cortex, and as we can see, leaving thalamus, signals will then go to the cortex, and we're traveling that signal to the primary auditory area specifically, which happens to be in our temporal lobes. Now let's look at how we stimulate our cochlear hair cells. Okay, it once again begins with the sound waves reaching the auricle. The auricle is just a fancy way to describe your external ear, what you can see. Okay. The sound waves travel inward and strike the tympanic membrane, causing it and the malleus incus and stapes to vibrate. And this causes the membrane of the oval window to vibrate, which then causes the perilymph and the body labyrinth of the cochlea and the endolymph, the membranous labyrinth of the cochlea, to move. And I'm not really going to get into it in great detail what the perilymph and endolymph are. They're just basically fluids on the inside of our cochlea that will ripple, if you want to think of it that way. I always think of the movie Jurassic Park. I know some of you guys probably don't remember that movie or never saw that movie, but there's a scene in that movie where there's a glass of water sitting in a tray that all of a sudden starts to see ripples, because there's a T-Rex walking and creating these ground tremors. Okay. Well, the sound waves that run into our inner ear create these kind of ground tremors, if I put it in kind of quotes. Okay. They create this ripple effect, so the fluids start to kind of shake. Okay. And so this movement of the endolymph causes the basal or spiral membrane to vibrate, which in turn stimulates the hair cells on the organ of Corti to transmit nerve impulses along the cochlear branch of that vestibulocochlear cranial nerve. Now, you might wonder, how do we detect whether it's a high frequency sound, a medium, or even a low frequency sound? Well, as you can see here, we're going to end up transmitting that signal to different parts of that primary auditory area in our cerebral cortex. And that is due to the fact that the sound waves strike different parts of our cochlea. This is a very simple representation of our organ of Corti. Okay. The higher the frequency, they're going to stimulate hair cells more near what we call the round window, whereas the lower frequency will stimulate hair cells near the beginning of the cochlea. Therefore, the location of peak stimulation of the hair cells allows the brain then to interpret the pitch of sound. And so that's why you can tell a difference between a baritone and a soprano. Here's an illustration of the sound or hearing pathway. I should say not sound pathway, but hearing pathway. Begins with the sound waves traveling in our external auditory canal, our ear canal. And that is actually after the sound waves have traveled past the auricle, our external ear. They strike the tympanic membrane, our eardrum. Our free bones then transmit that signal inward to our cochlea. This is a very simple illustration of our cochlea. Okay. We see movement of the perilymph, scala vestibuli, vestibular membrane, endolymph in the cochlear duct, basilar membrane, and then finally hair cells against what we call the tectorial membrane. The hair cells bend, specifically the stereocilia, which creates a receptor potential, which then creates a nerve impulse. And it's a lot of very complicated things occurring. What I really want to talk about, though, is what actually happens to the hair cells when they are stimulated. Okay? So let's look at how this transduction occurs. Transduction of the hair cells from mechanical vibrations into electrical signals involve those little microvilli, what we called stereocilia. Stereocilia was the plural form of stereocilium, which is what's written here. Okay. Notice that they're of different size or length, if you will. Okay. And connecting each one of those to one another is a tip-link protein. Okay. And they connect to a mechanically gated ion channel and the neighboring cell, or the neighboring cilium. Okay. As the stereocilia bend in the direction of the taller stereocilia, as we see here, the tip-link proteins will open ion channels, which allow cations, like potassium, as we see here, into the cell. Therefore, that cell is going to depolarize, which creates a receptor potential, which then triggers the opening of calcium channels, allows calcium in, and we know calcium then moves those synaptic vesicles towards the membrane to then release it by exocytosis, releasing neurotransmitters that will then synapse with our first order sensory neurons. And then that causes the signal to be then transmitted from the hair cells into an action potential in our first order neurons, which will then travel through the various ways that we just talked about. Okay. Now, when the stereocilia tip the other way, they bend towards the smaller or smallest stereocilia. Those ion channels get closed, which causes hyperpolarization, decreasing the frequency of nerve impulses in the sensory neurons. So when we are transducing sound waves, this is what we're doing. When we're not, this is what we're doing. Okay. It's kind of a pretty cool little process. Now, our next special sense is equilibrium. Or just balance. This also takes place within our inner ear. It does not involve the cochlea. It takes place within our semicircular canals or ducts and a structure called the vestibule. And this involves the vestibular branch of that vestibulocochlear nerve. The function of equilibrium is to maintain coordination, balance, and orientation in a three-dimensional space. The same type of cells are involved in this special sense. Hair cells with microvilli. Okay. Now there's two different forms of equilibrium, static and dynamic. Static is equilibrium, or static equilibrium is the perception of the orientation of the head when our body is stationary. What do I mean by stationary? Like not moving. Okay. The structure involved with this is called the macula. Okay. Specifically the macula saculi, lies vertically on the wall of the saccule, and the macula utriculi, which lies horizontally on the floor of the utricle. Changing our head position produces a change of pressure, stimulating hair cells that in turn stimulate receptors of our vestibular nerve. Dynamic equilibrium is where we're moving, which we're saying here as motion or acceleration. Can be linear, like straight forward, or angular to the side, or even in a spinning manner, which can be really, or put us really in a bind for our equilibrium. Okay, we all know if we spin around a whole bunch, we're going to lose our balance. And the structures involved in this, macula, which, or linear is perceived by the macula, angular is by the crista. A couple structures that we really haven't talked about too much. Transduction of equilibrium involves the stereocilia located on the surface of the hair cells, depolarizing and hyperpolarizing in the same way that we just talked about with our cochlea, in the hair cells for sound. These synapse with first-order neurons in the vestibular branch of the vestibulocochlear nerve. And then from there, the axons of the sensory neurons in the vestibular branch synapse with sensory neurons in the vestibular nuclei on the medulla. As we show here, we're showing more of the green coming into. Here's our medulla. There's our pons. Vestibular nuclei also receive input from our eyes and the proprioceptors. Okay, remember proprioceptors are the ones in our joints and in our muscles and tendons. Vestibular nuclei integrate information and send out signals that control movements of our eyes, help control our head and neck movements, control muscle tone in order to help maintain equilibrium. Other neurons will carry the signals to the thalamus from the vestibular nuclei. We even see it crossing over from the thalamus. Remember, that's our gateway. We then send the signal to our vestibular cortex, which is in the parietal lobes, providing us with our conscious awareness of our position and movements of the head and limbs. A very, very important special sense that we all probably kind of take for granted. Okay? Unless you ever have an inner ear problem and you lose balance a lot, which does happen from time to time, or when you get dizzy from spinning around, something like that. But for the most part, we really probably take for granted our just ability to maintain balance. Over here on the left side of the figure is showing you what, or left side of the page, showing you what happens when we change position. Here's our macula. The hair cells are located in here. The little microvilli or stereocilia are extending off of it. When they bend, as you can see, when we change our head position, they're going to depolarize, and that's going to create the whole process of an action potential. So, nothing really image-wise that I'm worried about here, but I wanted to present it to you anyway. All right, and our final special sense is vision, which I would say is the most complex of our senses. Our eyes, we all know, are the structures that allow us to see, something that we learn at a very early age of life. Our eyes are very complex structures. More than half of actually our sensory receptors in the human body are located in our eyes. And a very large part of our cerebral cortex is devoted to processing our visual info. The layer of the eyes where we have the receptor cells for vision is the retina. We call the receptors for vision photoreceptors. And these are the cells that absorb light, generate a chemical or electrical signal. And it includes what we call rod cells and cone cells, or what we just a lot of times will call our rods and cones. I'm sure you've probably heard rods and cones before. They get their name because of their shape. Our rods are responsible for night vision, and that's great. They don't distinguish colors from each other. Each retina has about 120 million rods, which is a very significant amount. Our cone cells function in bright light, responsible for day vision, as well as color vision, and each retina has about 6 billion cones, so a lot less cones than rods. Okay. Now there are other cells that are located in the retina that aid the rods and cones of vision. So we will call these non-receptor cells, or non-receptor retinal cells. We have what are called the bipolar cells, named because they are bipolar neurons. Okay. They're going to function to provide a large amount of convergence, stimulate what we call ganglion cells. These are the ones that receive the signals from the rods and cones. Ganglion cells are the cells that form our optic nerves. Optic nerves are, remember, cranial nerves too, connecting directly to the back of the eyeball. Okay, they're the ones that receive signals from the ganglion cells. And then we've also got horizontal and amacrine cells, which form connections between other cells and enhance perception of contrast, edges of objects, and changes in light intensity. Here in our figure of this next page shows you what the retina looks like from the inside. Okay. There are several layers to a retina. You have the pigmented layer, the photoreceptor layer where you find the rods and cones, the bipolar cell layer, which is where you find the bipolar cells. You also find the horizontal cells, and the endocrine cells. And then the ganglion cell layer, which is where we have the ganglion cells that then come together to form the optic nerve. Okay. We can see kind of the cone shape to the cones and the rod shape of the rods. This is where they get their names. You can see that these are bipolar neurons with the cell body on the inside of the two forks or branches on both sides. Okay. These cells synapse with the other cells that we talked about. We're converging all together to exit from several rods and cones to just a few ganglion cells. So we're kind of converging it all together. And then from there, the signal will travel out the back of the eyeball through what is known as the optic nerve. Now, to make matters even more complex, some of what we see in our eyes crosses over, but not everything. Okay, if you were to draw kind of an imaginary line vertically, splitting your eyes into equal left and right sides, the side closest to your nose, okay, what we call the nasal half if you wanted to, that is going to cross over. Okay. The retina or cells that receive that signal are going to cross over from one side of the eye to the other. Okay. Whereas the other side does not. So pretty amazing that with, you're just looking with your right eye, some of what we're seeing is being processed in the left side of our brain, some of it's in the right side of our brain. Okay. The optic chiasm is the point where the two optic nerves come together and cross over. The word chiasm actually means crossing over. And then from there you have the optic tract to the thalamus. Remember, there's our gateway. And then from there, the primary visual area of our cerebral cortex and our occipital lobe receives the signal. This just really demonstrates how complex vision is. But, the way that we process what we see is super complex. Okay, a couple years ago, the blue and black dress was a internet sensation. Some people saw it as blue and black. Other people saw it as white and gold. Okay, it has a lot to do with how we interpret what we see. Okay, there's a lot of variability between people with regard to seeing, and just the act of light and dark is very complex. Okay, the book talks about bleaching and that kind of stuff, but what happens when you are adjusted to dark light and then I walk over and flip the lights on? It's going to be hard to see for a couple seconds, almost probably a minute or two, because it takes a lot of inner workings of our eyes to really just function the way that they do. So vision is pretty amazing, also very, very complex.